So we're going to start off with glycolysis questions. So we have number 11, which says the intravenous infusion of fructose into healthy volunteers leads to a 2 to 5-fold increase in the level of lactate in the blood, a far greater increase than that observed after the infusion of the same amount of glucose. So why is glycolysis making more lactose or lactate in the blood after fructose? And why is it more rapid? And why is feeding fructose instead of glucose unwise? So, as you know, I didn't draw this part out, but when you have the pyruvates at the end, if you were to look at the fate of the pyruvates slide on the review, that wasn't there for, but I made the PowerPoint. You can see that pyruvate can either become acetyl-CoA in an aerobic environment and then enter the TCA cycle, or it can be converted into lactate, which is what's mentioned here, and that usually happens in an anaerobic human, or in yeast you have it turning into ethanol. But nevertheless, if you have more lactate, that means you have more glycolysis going on, because glycolysis happens, pyruvate forms, and then pyruvate turns into lactate. So the more lactate you have, the more pyruvate you have, the more rapid glycolysis. So it's asking you why there's more lactate when you infuse fructose instead of glucose. So there's actually a whole fructose pathway as well. So let me draw that. So here's a pathway for fructose. It'll be converted into fructose 1-phosphate, which will then be split into glyceraldehyde and DHAP, and each of those will be converted into G3P. And then once you make G3P, these two G3Ps will then directly feed into the glycolysis pathway. And so we basically see a bypass of this whole step, the first whole part of glycolysis. So it's asking you why it's more rapid. Well, it's more rapid because, as you guys learned, there's a very, very important enzyme in the first part. Remember what it is? It's phosphofructokinase. And phosphofructokinase is probably the biggest regulating enzyme in the entire glycolysis pathway. So I'll read more into that if you have any questions. And basically it manages the speed of glycolysis, so it prevents it from going too rapidly. So since it's bypassed, for B, why is it unwise? Well, it's unwise to feed fructose because then glycolysis will be unregulated. And so here we're making lactate, and so you would have an overproduction of lactate, and that would then cause lactic acidosis, and so that's kind of, you shouldn't cause lactic acidosis into healthy volunteers. So that's basically the gist of 11. So for 12, I'll understand what's going on here, but basically you have a biochemist that's studying glycolysis, and she's specifically studying glycolysis leading to ethanol. So if you remember in the last problem, that's what happens in yeast cells, and it's one of the fates of pyruvate. And so what she does is, when she goes through glycolysis, she makes the C3 position in the culture, she makes it radioactive. It's a radioactive carbon. So then she goes through the entire glycolysis pathway, but then none of the alcohol or the ethanol that you have at the end, none of it is radioactive. So she's wondering what's really happening. What's really happening with this? Why it's not radioactive? So if you look at figure 18.21a, it actually shows this very reaction. Or sorry, 18.1. I'm not sure. It mentions both figures. But essentially, if you follow the C3 carbon of glucose, then you find out that basically it ends up right here. This is the radioactive carbon. Just to label, this is pyruvate right here. This is pyruvate. So then what happens is pyruvate, when going into that ethanol reaction, a CO2 is cleaved off. So this, and this is done by pyruvate decarboxylase. So that's cleaved off, leaving this right here, which is acetaldehyde. Probably pronounced that way wrong. And then acetaldehyde goes through alcohol dehydrogenase. That's an R. Dehydrogenase. And that will finally make ethanol. So, essentially what's happening, if you've noticed, is that very first reaction, if you follow, this is C3 this is the C3 in the original glucose. If you follow that reaction, then C3 is radioactive, and so it will be lost in this pyruvate decarboxylase reaction. This whole thing is cleaved off, the CO2. So that's why in the ethanol, you don't see any radioactive carbons. So for 13, it says the following substrates can all be metabolized by or fed into glycolysis and converted into lactate. So how many ATP molecules per molecule of substrate are made in each case? So basically, if you put each one of these through glycolysis, how many ATP molecules will yield? So the first question is with glucose 6-phosphate. So if we just put in this glucose 6-phosphate, then you're gonna expend 1 ATP here, and then you'll make 4 ATP here. So you'll have a net of 3 ATP made. If you have fructose, then fructose you're losing 2 ATP to make the 2 G3Ps, but then with the 2 G3Ps you'll make 2 ATPs. So you lose 2 and then you're losing 2 and then okay, I see what I did. My bad. So I actually forgot to put one more ADP to ATP step right here. I was really confused as to why that wasn't adding up. But yeah, here you're going to be going from ADP to ATP. Sorry about that. Luckily, it hasn't mattered for any of the problems we've done so far. Okay, now it will make sense that when we go through G6P, then we're going to expend 1 ATP, and then we're going to make 4 ATP. So you're going to have a net total of 3. With fructose, you're losing 2 ATP, and then you're making 4 ATP. So you'll have a net of 2. If you look at sucrose, well, sucrose is a disaccharide made of glucose and fructose. So if we look at the glucose part, then you're losing 2, you're making 4, so you have a net of 2. And if you're looking at the fructose, like what we just did, we saw that we lose 2 and then we make 4. So then we have 2 again, so 2 from each. So you'll have 4 total. Next one's a bit tricky, dihydroxyacetone phosphate. So you would insert that here and here. So here, it will go through 1 G3P, and 1 G3P makes 2 ATP. And so, JK, that wasn't tricky at all. Yeah, so never mind. I think it was tricky because at first I thought that only 1 is made here, but the answer is 2. So I was like, where's the other one coming from? But yeah, there are actually 2 made in each of these. My mistake. So that's dihydroxyphosphate. The next one is glycerol. So with glycerol, it actually has its own mechanism. I'm not entirely sure if you guys have to know this for the exam because I don't remember learning this. But maybe it's changed. So let me just draw out the mechanism for you. First you have the glycerol, and the glycerol will then, I'll just write the substrates first. It will be converted to glycerol 3-phosphate. And then it will be converted into dihydroxy acetone phosphate, DHAP. And then that will be converted into glyceraldehyde 3-phosphate. So the first one is the enzymes doing it. Here you'll have the glycerol kinase or glycerol 1 kinase. I'm not sure if it's a 1 or L, but I think it's glycerol 1 kinase. I'll double check that. And then you have from glycerol 3-phosphate to DHAP, you have glycerol 3-phosphate dehydrogenase. And then from DHAP, you have triose phosphate isomerase. And that'll make G3P. So you actually lose, as far as energy stuff goes, the first step, you convert an ATP to ADP. And then the second enzyme, you'll convert two NADHs to, or an NAD plus to an NADH. I don't think this part's that important. You just have to look at the ATP more. See if you don't lose, you gain NADH. So in this reaction, if we put in one glycerol, then in that glycerol, you'll be using one ATP just to make the G3P. And then, each G3P, makes two ATP. So you'll lose an ATP here, and then you'll make two ATPs here. So you'll have a net of one ATP made with glycerol. Glyceraldehyde 3-phosphate is the next one, so you're just putting in one of these, so that'll create two ATP. Last but not least, you have galactose. So galactose actually goes through its own pathway. So I'm not sure if you have to know this pathway. I didn't. I don't think it'll be very high yield, but let me just draw that out for you. So here's the pathway for galactose. It will be converted into galactose 1-phosphate, which will then be converted into glucose 1-phosphate, and that'll be transformed into glucose 6-phosphate, and glucose 6-phosphate will then enter glycolysis right there. So you use one ATP just to make the glucose 6-phosphate, and then you would use another ATP here. So you've used two ATPs, and then you make four, so then you just have a net of two ATPs made for that one. So in 14, you have arsenate, and in the reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase, arsenate can replace phosphate in attacking the energy-rich thioester intermediate, and the product is unstable and will spontaneously hydrolyze to 3-phosphoglycerate. So what is the effect of this arsenate on the energy of the cell? So with arsenate, this is the reaction that essentially happens. You have G3P, glyceraldehyde 3-phosphate, and then you add NAD+, but then you have H2O, and then that will yield 3PG plus NADH plus 2H plus. All right, so in this, usually when you go from G3P to 3PG, you also make an ATP, but with this reaction, it has become absent, and so now you lose two ATP in glycolysis here, and then instead of making four ATP, you only make two ATP. So essentially, glycolysis doesn't yield any energy in the presence of arsenate because the ATP yielding step, one of the ATP yielding steps has been bypassed here. For 15 and 16, which go over gluconeogenesis, I figured it would just be easier to email you guys about that one. But here I've gone through, in this video, I've gone through 11, 12, 13, and 14. So good luck on your exam, guys, and just do your best.